I must acknowledge our chairperson, Steve. Thank you for giving us an opportunity today with this particular talk that we're having. And I must welcome AP. It's nice to see you again. And of course, our past international president, Barry. My role today is to introduce you to Professor Mark von Itzstein, AO, BIO. Our foundation, your foundation, the Lions for ALCCRF, in 1921, the trustees of the Australian Lions Child Cancer Research Foundation approved funding for the Griffith University Institute for Glycomics project that focuses on sarcoma. Today, I have much pleasure in introducing the team leader of that project, Professor Mark von Itzstein, AO. Professor Mark is a fellow of both the Australian Academy of Science and the Australian Academy of Health and Medical Science, and a joint recipient of the prestigious Australian Prize in 1996. He was also appointed as an Officer of the Order of Australia in 2019 and has won numerous national and international awards during his career. Professor Mark led the team responsible for the design of synthesis and biological evaluation of anti-influenza drug Relenza, which has been approved for the treatment of influenza worldwide since 1999. This discovery is considered to be a significant outcome and flagship in glaucoma therapeutic and antiviral drug development in the last century. Professor Mark is the director of the Griffith University Institute for Glycomics, which is the only one of its kind in the Southern Hemisphere and one of a few in the world. The Institute's research collaborates with leading scientists around the world, and it's a global, it's a global project. And today, Mark is here to give you a lot of information on where we are with his projects and what it's actually, the aim of the project is to do and the outcomes. And he's had so many publications. I think the publications are going to about 240. And so I'd really like to invite Mark to come up here today and to give you a really good idea of what your foundation and you have been doing. So I welcome you to the microphone, and I'll turn it over to you and you can explain those other words that I couldn't get out better.
Thank you, Tony, for your kind words of introduction. Yes, glycomics is not a trivial term, I must admit, and certainly some of the terms that Tony was rehearsing are also not so trivial. Look, may I first of all thank you for the kind invitation to come and present today. It's a rarity that I have so many people in one room from a fantastic organization like Lions International. And if you know me at all, I love talking. And I love talking passionately about science, whether it's our science or science anywhere in the world. Because at the end of the day, it's our only hope in fighting diseases. Medical research is our only hope in fighting diseases. And so today I thought what I would try to do is not drill down on the science, but talk to you a little bit about my institute, what we established here, right on here on the Gold Coast, actually. So it's not just thongs and singlets and budgie smugglers. There's very good scientific infrastructure. And indeed how we connect internationally as well with the world and trying to solve intractable diseases. So cancers, as we know, is certainly an area where you'd think after how many years have us medical researchers been working in the space of cancer, and still whilst major advances have been made over a number of years, and we know that, we appreciate that, we still do not have a series of magic bullets that says that is now a thing of the past. So for me, that speaks to, and I was saying it a little earlier on, it tells me that whilst we are making major advances and major breakthroughs, there's still a few pieces of the jigsaw puzzle that we haven't brought into focus. We need to bring that into our lens to be able to combine it with all of the other knowledge that we collectively as scientists have discovered. Now, I didn't educate myself on starting the slideshow, so do I do that or do you do that? Could somebody start that slideshow for me, please? I see it there, but I don't see it there. It's only on there? Brilliant. Okay, thank you for clarifying that. I can see it here, so that's okay. I imagine that I am doing this. Brilliant. So the Institute for Glycomics was established in the year 2000. I moved up from the deep south, from Melbourne, to specifically establish this institute, because in this country, and indeed in the Southern Hemisphere, and in fact at that time in the world, there was no single established institute that brought the disciplines of science together, chemistry, biology, physics, computational science, under one roof to tackle diseases from the perspectives of glycomics. And I'm going to tell you what glycomics is in a short second. So that's what we established in the year 2000. When I moved up with about 5 people. Now there's over 200 folk in my institute in the year 2023. So this is where we are on the Gold Coast, and isn't it beautiful weather today? I didn't think it was going to be like this outside today, but I'm so pleased for our visitors outside of Queensland that it is beautiful weather. So right here on the Gold Coast, what we have established, sorry, I keep on looking there. What we have established, as you can see there, is a health and knowledge precinct that wraps around our university, that's Griffith University, with the 2 major hospitals that we've got in the same precinct, as well as if I call it like this, an industry park, where we attract in various industry partners, and it's growing very significantly. And that co-location of those assets in medical research is phenomenal. Why? Because we have the capacity of working then with clinicians. I'm not a clinician, I'm a simple PhD scientist, but I work with many clinicians. We have clinicians in our institute that are at the pointy end dealing with patients on a day-to-day basis. And so bringing all of those fine folk together into a room to tackle diseases, broadly speaking, really accelerates outcomes. We don't want a discovery in 50 years' time. We want it within 10 years, right? We really want to be advancing science. And so my education led me to believe that bringing teams of researchers together that tackle things from the basic science point of view, those folk that are actually translating the research and working with clinicians to go into human clinical trials, and eventually industry, because we need industry, obviously, to push it to market, the drugs or vaccines that are being discovered, is a good thing. It's a melting pot. It's a melting pot. And it's a great training ground for our researchers, our young researchers particularly. So we're right here on the Gold Coast, and in fact, only 15 minutes away from this site right here. If you have the chance of visiting the campus, if you do, please do. If you want to come to the Institute, let me know. I'm more than happy to show you around. So our vision right from the early days was to establish a vibrant and preeminent glycomics research institute that was specifically geared towards delivering new diagnostics, new drugs, and new vaccines to treat a range of conditions, particularly diseases that are intractable, both in the infectious disease space as well as in cancer. So as I mentioned earlier on, starting from a handful of folk that were brave and moved with me from Melbourne to the Gold Coast. We now have over 200 clinical and basic research scientists in the Institute. And what is really nice after 23 years having established this institute, we really now have got international traction as well as national traction, where folk that are interested in the way we tackle disease problems come to us, irrespective of the country, from China, from Europe, from the US, Victoria, New South Wales, etc., and the Latin Americas as well, because of our expertise, which is unique in the country. Far too often I get the question, what's your point of difference? Why are you different to the Walter and Eliza Hall in Melbourne? Why are you different from QIMI and Berghofer in Brisbane, etc.? And I'm always delighted to be able to say these are the institutes that we work with, and as well as others, because we have a unique expertise in this nation. So we're not duplicating. That's critical, because you find folk that are investing, and through your foundation, you've invested in us. That's how we look at it. We don't want to waste 1 cent of the money that's provided to us from such a foundation and duplicate some other work that somebody else is doing in the country. We want to make sure every dollar that you invest is bang for buck. Our translational research capacity is critical as well. So within our institute and on our campus, we have the capacity to do discovery science, as you can see there, discovery research. So we have facilities that are oriented in that space that then enable us to do preclinical development, so we can actually take our vaccines and drugs into the next level of a bioresource facility to better understand how well do these things work in animals before we march into, as you can see there, manufacture on the occasion of biologicals like vaccines. We can manufacture on campus, and we've got a malaria vaccine candidate we manufacture on campus, and then we put it into humans in a phase 1 facility. That's rather unique in this country, but we purposefully did this because we wanted to be a translational research institute. We wanted to make sure that we deliver new medicines. So this is Glycomics.
We think of sugars or carbohydrates. We often think of the simple kinds in our diet, like glucose and starch. But did you know that every cell in our bodies is decorated with a vast and complex array of sugars? These sugars on our cells are crucial for cells to communicate with one another and the environment. Our bodies need these sugars, known to scientists as glycans, to stay healthy and to fight disease. In fact, glycans have been found on every type of known organism on the planet. We can think of glycans as a type of sugar language that our cells can use to send and receive messages. Without this sugar language, the human body would not function. What many people don't know is that many diseases take advantage of the glycans in our body, resulting in major health problems and sometimes even death. At Griffith University's Institute for Glycomics, our scientists are working to better understand the role of glycans in the development of diseases so that we can better diagnose, treat, and prevent diseases such as cancers and diseases caused by deadly pathogens such as parasites, bacteria, and viruses. Let's take a virus like influenza, which causes the flu, as an example. The influenza virus uses a protein on its surface to latch onto glycans. The flu can become dangerous when it spreads to tissues in the body, and that's why it's important to treat this virus as early as possible or prevent infection completely. Proteins on the surface of the virus latch onto glycans. Once bound, the virus then infiltrates the cell and begins to replicate. Virus progeny then emerge from the infected cell and seek to infect other nearby cells in our body, ultimately overwhelming the body with infection. Researchers at the Institute for Glycomics focus on the role that these sugars play in disease, and using that knowledge, invent new drugs, vaccines, and diagnostics to treat, prevent, or diagnose diseases in the hope to save millions of lives globally. The Institute for Glycomics, fighting diseases of global impact.
So glycomics is the study of sugars, particularly as it relates to diseases like cancer. And so I'll switch directly to cancer now for a second. Over the past, I guess now 25 years or more, we have been playing detective to try and understand how healthy cells present these glycans, these sugars. And indeed, when cancers appear, irrespective of the cancer, the changes that occur, and changes do occur. Changes occur from different stages of tumors as well. Understanding how those changes are occurring gives us opportunities to look at the discovery of novel drugs, because we can look at new drug targets. We can also think about actually developing a vaccine against a particular glycan that is unique to that cancer signature. So a lot of our efforts really are looking hard at the makeup of those glycans and the changes that occur when cancer, unfortunately, is established. So these are very nice cartoon type slides, which I quite commonly use. Yeah, here's a real image now of what the glycans, the sugars, look like on cells. So you see that nice circle there? That nice circle is actually a blood vessel. And you see these black protrusions coming out from the surface of that blood vessel. That black matter are all sugars connected to the cells associated with that blood vessel. So when you look at that and you see that nice rectangle, red rectangle image that brings up that sugar layer, that candy floss layer, you can see it's enormous. So when you think about, for example, cancer cells that are metastasizing, spreading around the body through typically blood vessels, it doesn't take too much to think, well, they must be continuously coming into contact with those glycans. So when the tumor cell that's moving around the body wants to lodge somewhere and then grow again at a secondary site or a distal site, as we commonly term it, it has to work out how to latch onto those glycans and then move into the new environment and start new blood flows, etc. So understanding those mechanics, those processes, gives us the opportunity of saying, how do we stop it? How do we stop actually that cancer cell moving around and lodging in a new domain? So that's what we do. So you could imagine that the technologies then that are necessary to decipher that language, to decipher that code, to break the code of cancer are pretty intense and very, very costly. So in the Institute, we have a range of cancer programs going on right now, some that we've already published in, particularly in leukemia. I'm not going to dwell on the detail there, but you can see that interesting colorful map there. That colorful map actually is informing us about sugar changes in leukemia cells, particularly acute lymphoblastic leukemia on this occasion, a childhood cancer. And understanding those changes gives us valuable insight, again, to identify new targets to tackle acute lymphoblastic leukemia, particularly when children go into remission. So that study that we published, there is a reference there somewhere, that we published a couple of years ago now, really informed us about what's happening to make that particular cancer drug resistant when the child unfortunately comes out of remission. So when a child typically gets leukemia, mostly there's a good cure available. It's when they go into remission and then fall out of remission is where significant loss of life occurs. So we're particularly focused on drug-resistant tumors on that occasion. Glioblastoma, so a lot of brain tumors, as we've been hearing in the news more recently with childhood brain tumors a big issue. And so we've got now technologies that we can intimately look at, almost like looking through a microscope at the glycan changes in these brain tumors. And as was mentioned by Tony in introductory remarks, sarcoma is where the foundation, Lions Foundation, has really helped us in establishing us profiling sarcoma, particularly childhood sarcoma, and that's what we're doing right now. Sarcoma is very much an underestimated killer. And what you may not appreciate is, in fact, as you can read there, is that there is over 70 types of sarcoma. Of course, we can't tackle 70 types of sarcoma in one go. So it's a very important childhood cancer. So as you read there, over 20%, in fact, of childhood cancers are indeed sarcoma. And the real issue is that this is a cancer that's almost for every organ in the body, and that's scary in lots of ways. It can start absolutely anywhere. And so this is just now some of the soft tissue and the bone sarcomas that are known. So you can see it can actually commence anywhere. So that's pretty scary. It's a cancer for every organ virtually. So the problems, here's the challenges that are being faced right now. Lack of early detection capability. That's something we need to be able to solve rapidly. In fact, we hope in a handful of years through the support of the Lions Foundation. The clinical management of, in fact, patients that are coming in with sarcomas. And the big problem is that it's such a mixed bag. The heterogeneity, we term it the heterogeneity, the mix of cell types in sarcomas is very challenging. And indeed, as you can read there also, the mere fact that typically these tumors can be very insensitive to current treatments. So what we're trying to do, of course, then to address those challenges is to try, as we do for every cancer, try to get an early blood diagnostic that says, okay, we know not only that you've got sarcoma, but what type of sarcoma you've got, which then we hope will aid us in being able to inform the clinicians about the personalized treatment of an individual that will hopefully assist them in treating that particular sarcoma in that particular patient. Importantly, this is an international project. So we formed an alliance, as you must do. You've got to work with the best. So you know that I'm not a clinician. So we have to work with those fine pediatric clinicians that are actually dealing with those patients on a day-to-day basis. So we have a hospital in Uruguay, and many of you would know the Chris O'Brien Lifehouse in Sydney that has a very fine program in sarcoma. And so the interplay, again, between the basic science and those clinical researchers is critical for us to be able to work together to find the solutions that we desperately need to find. Just checking on my time. I've got a few minutes left, so that's good news. Otherwise, I know what I'm going to get. Okay. So we take that integrated approach very, very seriously. There's no point in us basic scientists running off in one particular direction whilst the clinicians are thinking something quite different. So working together, we hope we can address the challenges that I just mentioned on the previous slide. For your information, the projects that we're starting with in sarcoma are with rhabdosarcoma and fibrosarcoma, both very significant sarcomas in kids, unfortunately. You can see rhabdo is more hitting the muscle, and on this occasion in the arm, and fibrosarcoma in the bone domain in the context of fibroblasts. And so these are very prevalent in pediatric populations, certainly prevalent in the collections that we have historically available to us now through Chris O'Brien Lifehouse, and the Perez Scremini in Uruguay, but also the ongoing collections from new patients that are unfortunately coming in. So my researchers, I'm the director of the Institute for Glycomics, so my job is to raise money to support the research that my fine and talented researchers are doing. So the kit that we've now bought over many years looks something like this. Yeah, you can see it there. And proudly sponsored, by the way, by the Australian Cancer Research Foundation. We won a major grant to get the equipment that we needed to do imaging through that microscope lens of looking at various cancer cells. So it's a unique facility in the world. It's important to tell you it's a unique facility in the world in the collection of apparatus and the talent that we've got, and is highly used by individuals right around the world. So we have many collaborators through Europe and the US that send students and staff for training to work with us in imaging their various cancers. So here's a DIPG you may have heard of in the news very recently. It's a brain tumor, unfortunately, in young kids. And so we've got a major program. And so I just didn't want to go into the detail here, but this is the imagery that we see in those beautiful colors. So we get a biopsy on a slide, as you can see on one part there. We process it through a series of techniques and then effectively put it on this fancy microscope. It's not a microscope, it's actually a mass spectrometer. I won't go into the detail about that. And we get images like you see here with very intense colors. And we can interpret those intense colors because they're associated with particular glycans. So we can actually map where the tumor is, what stage the tumor is at, and how we might actually tackle that. So you can see a reference that's all blue, there's no tumor. In the middle, you can see where there is tumor present, and then post-treatment, if I recall rightly. So the technology that we have available now, compared to even 5 years ago, tremendously different. So when we say, yes, we can find out new information, you can believe that we can find out new information that we know will work very well with other bits of information that come in from other areas. You've all heard of genomics, I'm sure. So genomics, as you know, revolutionized our capacity to understand changes in cancers, even in drug-resistant cancers. So there's information that's coming all the time. What we do now is that we multi-layer that information of genomics with our glycomics and proteomics and lipidomics. And that's the modern way of the world in medical research, that you are taking all of that information and getting a holistic view of tumors. And remember, I did start by saying that it's surprising in many ways that we haven't really solved cancer yet. And there must be missing bits in the jigsaw puzzle, pieces in the jigsaw puzzle. And so we believe that that multi-layered approach in bringing all of the information together will lead to fantastic outcomes. And that's what we want to do at the end of the day. We want to be able to diagnose patients, obviously, then with research and discovery, translating that into giving precision diagnosis and treatment for patients, on this occasion by support in the sarcoma sense. So that's the international team that we've now established. And you could imagine that it takes time in part to get human ethics approvals to start research, which are all in place now. So I'm very excited to say that the sarcoma project is well underway now. And so we are starting to see then, with ethics approval, the specimens arrive for us to start to play that detective work to give us the information that we need to say we understand what's going on in terms of the changes in those two types of sarcoma that I mentioned. So with that, I'm going to stop. I think I've made it on time. I have by 1 minute. I don't get a flower. Most importantly, I do thank you for your generous support, because without your support, whilst we win money from the government's agencies, etc., you probably don't know that 90% of researchers are not winning money because the budget simply isn't there. 90% of researchers are not winning money. And so there's a lot of people leaving medical research now as a consequence. I hope that changes in this country. Thank you.
Could I ask Council Chair Steve to come up?
Mark.
What a great presentation. It's great to see the value that our foundations are providing, not only to what we can do in support of our work here, in medical research in Australia, but how that reacts and sort of intercedes with the work that's being done around the world. And it's great to see the work that Professor Mark and his team are doing here right on the Gold Coast, and certainly working well with others. And that is in no small part thank you to the great work of our Australian Lions Childhood Cancer Research Foundation. And it's something that we should be very, very proud of. And as Lions, absolutely, we should continue to support it. And when you see that being done, why wouldn't you support it? So, Professor Mark, thank you for coming along and being our guest and speaking to us. Thank you for the great work you're doing. And thank you for the presentation today. Let's come over here, though. So, on behalf of everybody here, I'd like to present you with my banner as Chairman of Lions Australia. Thank you.